Okay, to discuss our topics in the midterm in microbiology lab. So we go now to the actions of microbial control agents, which causes alteration of membrane permeability, wherein there is what we call a destruction that will happen in the cell membrane of the bacterial cells, damage to proteins and nucleic acids, which lead to the death of the bacterial cells or prevent the multiplications of the bacterial cells. And then we have the discuss also the rate of microbial death. Okay, physical methods, we have moist heat, pasteurization, and dry heat sterilization process, the filtration, low temperature, desiccation, osmotic pressures, and radiation process. So, and we have also the types of disinfectants which will kill or inhibit the growth of the bacteria. Phenol and phenolics, we have chlorhexidine, halogens, alcohols, heavy metals and their compounds, surface active agents, quaternary ammonium compounds, and organic acids. So actions of microbial agents, uh, we have the alterations of membrane permeability, the damage to proteins and nucleic acids, and rate of microbial death. So actions of microbial control, so alterations of membrane permeability. So yung mga quaternary ammonium compounds, these are agents which can cause leak of the cell membrane. Okay, so the plasma membrane controls the passage of the nutrients or the entry of the molecules or food that enters in and out of the cells. So damage to the plasma membrane causes the leak of cellular contents and interferes with cell growth, because if there is a damage in the cell membrane parts of the bacterial cells, so other molecules or other toxic or even antibiotics or antimicrobial agents tend to freely enter the cytoplasm of the bacterial cells, and it will cause also damage to other organelles, and it will cause lysis and death. It will cause also uh, uh, what we call this one, damage to the genetic material of the uh, bacteria, which will inhibit its growth and even kill the or even it will lead to the death of, I mean, of the bacteria. So we have here quaternary ammonium compounds. Okay, so detergents have antimicrobial activity and are effective disinfectants. Okay, so based on the structure of that quaternary ammonium compounds, we have this cetylpyridium, and we have the benzalkonium chloride. Benzalkonium chloride, yung common name yan sa market is mertiolate. So most likely disrupts cell membrane, and they are considered to be a positive detergents, or cationic detergents are effective disinfectants. So it kill most of bacteria, but not the Mycobacterium tuberculosis or even endospores, because they are protected by spores, and these M tuberculosis are protected also with mycolic acid that is present in the cell wall. Okay, and this is considered to be very safe and safe to use. So it will inactivated by hard water and even soap. So this one, quaternary ammonium compounds, mostly they are considered to be antipathic organic cleansing agents. So when say antipathic, they have a water and the oil phase, because soap is composed also of mineral oil or oil. Olive oil is also used. I mean, okay, may ano yan, may oil na may fats. I mean, sorry, that may fats content ang yung mga cleansing agents. So sa disruption, they tend to leak the cell membrane, and leakage of that cell membranes, again, as what I have mentioned, the chemicals or other agents or other toxic agents can enter freely to the cells, and it will cause damage to different organelles of cells, especially the genetic material, I mean, to that will lead to inhibition of the of multiplication, I mean, or growth and multiplication, I mean, of the bacterial cells. Okay, So damage to proteins and nucleic acids. So mode of action of antimicrobial agents, there is alterations of permeability, so lipids and proteins. So when there is a leak, that leads to damage already of the nucleic acids or proteins. So damage of proteins, especially cellular proteins that is present inside, and nucleic acids, and it will lead to inhibit the replication process or multiplication, of the bacteria. So DNA and RNA carry genetic message, damage to them by radiation of chemicals frequently lethal to the cells, because it is lethal because when there is a damage of the RNA and DNA, so one important that will happen to the bacteria is the inhibition of the growth and even the multiplication, and even of course the bacteria will die. Okay, so rate of microbial death. So bacterial populations killed by heat or chemical tend to die at a constant rate. For example, 90% every 10 minutes, plotted logarithmically. These figures form a straight descending lines. Yes, of course, if we have the logarithm of the number of microorganisms or microbial, what do you call this one? Microbial survivors. So usually, based on the curve, so one log decrease 90% of populations. So the curve is plotted logarithmically, okay, and arithmetically broken time. In this case, the cells are dying at the rate of 90% each minute. So there is what we call a, a fast decrease, of the bacterial cells. And the effect of the high or low initial load of microbes, if the rate of killing is the same, it will take longer and kill the members of the target populations that's as one smaller. Okay? So kung marami yung bacteria, of course yung killing what we call this one, the dying capacity is also slow. So the death of bacterial cells or the rate of microbial cells, it depends upon the number of cells, okay, on the number of cells, that a number or population, of bacterial cells that is present in the uh, specific environment. So kung mahina siya, okay, kung gamay yung population load mo, madali lang yung what we call this one, the death. So mas dasig yung death capacity nila. Okay? While kung damo yung bacterial cells, of course, medyo mahinay man ang death capacity because it depends upon the populations. So kung if you give what we call this one, a drug, okay, same drug, same dose, but different quantity of bacteria, of course, if there's a large population, of the bacteria, so the death rate is slower kasi damo sila. Kung jutay lang yung bacteria, the same dose, example, ng drug na ihatag mo, so faster yung death rate ng bacteria. So gamay lang sila. Okay, so the microbial death depends upon the population of bacteria in a given environment. So kinetics of bacterial death. Bacteria not only grow exponentially but die the way too. Yes. Kasi the factors that affect the rate of death, not only the population but also the temperature. The higher the temperature, the faster is the what we call this one, faster yung death ng bacteria. Okay? Even pH. Okay? So it depends upon the survival of the bacteria. Diba may mga pH tayo na mga acidophiles, na what we call this one, mga mesophiles bacteria, diba? For example, concentration in disinfectant. So the lower the concentration of the disinfectant which is given in the larger population of bacteria, so slower man ang death capacity ng bacteria. So if there's a large amount of disinfectant and smaller population, of course, what we call this one, there is what we call a faster death capacity bacteria. But if larger amount of disinfectant plays in larger amount also of the bacteria, so there is a faster death capacity. So it depends upon the concentration of disinfectant, the quantity that is being used, type of microbes, and presence of organic material. So it depends on the type of microbes kasi ang type of microbes, there are a lot of microbes that will survive in different environment, in what we call this one, microbes that has spores or endospores which protect the entire cells for different exposure or different environment. Okay, presence of organic material also. So the longer the treatment is applied, the more that are killed, the more microbes that are longer it will take. Yes. So if you treat longer yung treatment, okay, of the antibiotics, the more that it will kill. So for example, that's why mayroon man tawag na microbial resistance, di ba? Remember that if doctor prescribes antibiotic good for 7 days or 14 days for example, then the patients will take it only for 3 days. So shorter yung treatment niya. So there is what we call a lesser capacity to kill the bacteria. But the longer treatment, when you finish of 7 days, then there is what we call a positive outcome that all that bacteria will be killed during the infection period. Okay? The more microbes there are, the longer it will take. So it depends also the populations. And that is why if there is infections, so the doctor will always prescribe 7 days. As pharmacists, we have to advise also the patients regarding antibiotic treatment. Okay? To prevent the resistance, of bacteria. Because when bacteria invade the body, it is comprised of millions. So that's why they can able to resist if the drug medication is not properly taken by the patient. Okay. So actions of microbial. So we have the physical methods of microbial control: moist heat, pasteurization, filtration, low temperature, desiccation, osmotic pressure, and radiation. So sa moist heat, it destroys also fungi, bacteria. So mostly, moist heat, ito yung autoclaving because it can have at least more than 100 degrees Celsius of heat that will kill bacteria. So boiling will not destroy spores. Yes. So spores are being destroyed at higher temperature, usually mga 1000 degrees, diba? Degrades nucleic acids, denatures proteins, and disrupt cell membrane. So especially the target of moist heat is to degrade the nucleic acids to prevent the multiplication. Okay, denatures proteins. So proteins is involved in the synthesis and even in translate and DNA translation. So ma-prevent rin yung multiplication, even it will cause the death. Okay, because yung mga nucleic acid or proteins are responsible for the control of the functions of the cells also and disrupts cell membrane. Okay, so pasteurizations. Louis Pasteur in the early days of microbiology found a practical method of preventing spoilage of beer and wine. So he used mild heating with sufficient kill bacteria that cause particular spoilage. So usually milk was first pasteurized to eliminate tuberculosis bacterium. Usually ito yung pasteurization. So it involves heating. So the heating is almost 40 to 50 degrees Celsius. Hindi siya masyadong boiling because it will cause or destroy already the fats that is present there. Okay, so if we have the meat coming from milk coming from the cow. Okay, so there's a heating section and cooling section, and the product is considered to be the pasteurized milk out. So pasteurization, eliminating pathogenic microbes, lowering microbial count, and prolong shelf life, or maintains the stability of the milk without altering the flavor. Okay, high temperature at short time. Okay, so lagay natin 140 degrees for 3 seconds only. So store several months without generation, or 60 degrees for 30 minutes, 72 degrees for 15 seconds. Okay, or in HTST is high temperature at short time. So and we have ultra high temperature, UHT. Okay, this one is considered to be 72 to 15 seconds. Okay, kasi kung i-boil mo siya 140, hindi naman siya kasi 3 seconds lang. So ma-prevent yung degradation ng fats inside, fats that is present or other nutrients that is present in the milk. So 140 degrees, for example, is just 3 seconds, and it can able to kill other kill bacteria also. So dry heat, one of the simplest method of dry heat is direct flaming or flaming. So that's why Based on our laboratory, we have to do the flaming of the inoculating loop or wire loop, okay, to prevent decontamination. Incineration, the way to sterilize, dispose contaminated paper bags, cups, bags, and dressings. Usually, ito yung mga hospital waste are being incinerated or undergone incineration process. So, sunugon siya, abono na pagwa. Hot sterilization, items to be sterilized by this procedure are placed in the oven. Okay? Temperature 170 degrees Celsius for nearly 2 hours. So, that is considered hot air sterilization process. Dry heat sterilization, it is effective and safe for metal instruments. Okay? So, oven yan. And then this is less reliable than autoclaving. So, many materials do not tolerate dry heat as media. Usually, ang dry heat kasi medyo mataas mo lang yung temperature. And then it is limited only for the metal instruments. Okay? Example, yung mga dull instrument edges or rust will going to corrode instruments. So before mo magkaroon ng dry heat sterilization process, so you have to check yung mga metals kung mayroon siya mga rust. So filtration is the passage of liquid or gas through the screen-like material with pores small enough to retain microorganisms for counting. So we have high efficient HEPA filter. So this is a bacteria filter, high efficient particulate air filters. Remove almost all microorganisms than other 0.3 micrometer. Using mga membrane filter, Sykes filter, Chamberlain, so Mander, yun yung mga filter na I mentioned in the prelim. So ito yung mga HEPA filters. HEPA filters is also used in manufacturing process, no? When you work at manufacturing, yung aircon, naka-HEPA filter yan. Kasi the air coming from, kung na-vacuum ng aircon yung air coming from the outside environment, so that air contains viruses or other microorganisms, so it will be screened, no, before it will be used up by the reconditioning. Okay, so kailangan may high efficient particulate air filters or HEPA filters, especially in mga sterile area. Okay, hospitals may mga HEPA filter yan. So low temperature, effects of low temperatures on microbes depends on their particular microbes intensity of applications. So decrease chemical reaction and possible changes in proteins. So if there is a low temperature, kasi in the chemical reactions, usually bacteria utilizes energy. So there is a use of heat. Okay? So if you lower the temperature, it will decrease the chemical reactions. Okay? Kasi sa chemical reactions, energy is being used up and there is a heat that is involved. So it has a bacteriostatic effect or inhibit the growth of the bacteria. So it is used in food, drug, and culture preservation. Desiccations. So condition of an absence of water. The state of extreme dryness or process of extreme drying. So that is desiccation. So vegetative bacteria resistance to desiccation varies with species of bacteria. So it depends on the species of bacteria. Example, yung mga Neisseria gonorrhea or gonorrhea bacterium can withstand with dryness for only 1 hour, but tuberculosis remains viable for a month. So it depends. Okay, kasi may mga iba may mga induced spores, they can survive at the different environment. Disruption of metabolism. So disruption of metabolism bacteria leads to slow the capacity of bacteria to cause infections because there is no energy that is being utilized bacteria during the infection. Bacteriostatic inhibition of growth and of course the food preservation are examples of this examples of the importance of desiccation process. So this is considered to be the desiccator. Okay, so usually what desiccator class usually there is what you call dehydration process that will happen that will cause the death of the microbes. Okay, osmotic pressure, high osmotic pressure cause plasmolysis or lysis of bacterial cells. So In high osmotic pressure, there is what we call a loss of water that will lead to lysis. So there is what we call list, what we call this one, parang mag-lyse yung cells, so there is what we call a death of cells that will happen. Usually it's preservation, it is used in preservation. Okay. So physical methods of microbial growth, we have radiation, so ionizing radiations. So wavelengths shorter than 1 nm or electron beam gamma rays. This is considered to be inject from the atoms to create ions. So ions will disrupt the hydrogen bonding, cause oxidation, and creates hydroxide ion. That oxidation process, okay, or the product of that one is the hydroxide ions will tend to denature other molecules, especially DNA. So that will affect already the multiplication of the bacteria. So hydroxyl group will bond with the DNA that will denature, okay, or will degrade the DNA, okay. Electron beams effective in killing but do not penetrate well. So gamma rays penetrate well but require hours to kill microbes. So yung radiation matagal lang, okay. So because it requires a long time, long hours to, to penetrate the cells, okay. And after penetration, so oxidation, so ions disrupt hydrogen bonding, okay, and oxidation, so there is what we call the formation of hydroxyl ion that will interact with DNA, and it cause denaturation of DNA already, okay, because there is already a changes in the sequence of DNA by the addition, of the hydroxyl group that will disrupt already the the normal uh, molecules, because nagbana yung hydroxyl ion, that is already alterations, okay. And we know that DNA is composed of amino acids, so that is a chemical, so that is considered to be amino acids are also unsaturated, so they tend to undergone processes that will bond with hydroxyl ion, and it lead to alterations already, okay.